electrophoresis is separate with the, today we are asking about gel electrophoresis. The gel electrophoresis is separate with the molecules on the basis of their charge and size. So the charge is macromolecules migrate across the span of gel because they are placed in an electric field. The gel is act as an excessive retarded passage molecules according to their size and shape. So one of the basic tools of modern biotechnology is gene splicing. So the process of removing the functional DNA fragments. A gene is from one of the organisms and combining it with the DNA of another organisms to study how the gene works. So the desired result is to have the new organisms carry out the gene expression and the gene that has been inserted. So how the restriction enzymes, the ability to cut the past DNA and predictably, so is due to the uses of restriction enzymes. The way first identified in isolated from the bacteria that use them as a natural defense mechanism to cut the pieces of invading DNA bacteriophage viruses that infected bacteria. They are named as for the negatively charged, the particles move towards the positively electrodes, while the positive charged particularly move towards the negative electrodes. So the electrodes, how the works, the gel is made from the agarose and the DNA is negative molecules short, a short based on the charged the base. So the agar is the major concept here. So agar is see wood it is work means DNA into the cell smaller fragments. Loading the dye is used to indicate the fragments of DNA behind the dye. So the negative DNA molecules is attracted to the positive electrode. The smallest fragments move the greatest distance. So then how this procedure will talking about this procedure means that remove the comb of observation wells and place carbons of preparations of the tray. So cover with the buffer and making the sewer allow the buffer and overflow. So then continue with this. These are the requirements of procedures. So how they are means the content with the loading gels and connect with the electrodes and turn on the power supply and allow the gel to make sure the see bubbles and coming these things. So then continues these things like a procedure. So that it will be take 30 minutes of gel run. So turn power supply and remove the electrodes and pour of buffer into the designated container. So carefully remove gel from the gel box and place into the glad container and covers with the stains. So store in appropriate locations. So finally we're talking about significance of these bubbles. So they indicate the electrolysis of water is taking place, and one electrode will have a lot of bubbles and the other will have a lesser amount. So why the difference? So the formula water and water is H2O, and the splitting of the molecules will be produced twice as many atoms of hydrogens. So like that we will talking about today. We will start talking about this. We will concluding this electrophoresis. So gel electrophoresis. So how DNA we will estimate it. So by using what are the procedures and requirements. So what are the functions of gel and then restrictions enzymes, and these are the things we will talking about these things.